Dr. Thamarai Janganan. Thamarai has worked as a clinical microbiologist and a lecturer. His research is in the field of antimicrobial resistance, looking specifically at multidrug efflux pumps in gram-negative bacteria.
Thank you. Thanks for inviting me to present my data. So thanks for introducing me. I'm basically a clinical microbiologist. I worked with a number of multidrug resistant clinical isolates, and then later I was interested, and then I moved to molecular microbiology, and then I was interested to look at how these bacteria, they develop drug resistance, especially how they export the antimicrobial agents out of the cell. Okay, today I'll be talking about MAT transporter in Campylobacter coli. So what is Campylobacter? So Campylobacter is a gram-negative. It's a curved rod-shaped bacterium, and it causes a disease, Campylobacteriosis. So Campylobacteriosis is like kind of gastroenteritis infection, and it's self-limiting in a normal person, and it's more complicated in immunocompromised individual. So what is the source of Campylobacter? So Campylobacter is a normal gut flora in chicken as well as in domestic animals, and the source of contamination is mainly poultry. So according to the Food Standards Agency, about 73% of supermarket chicken were tested positive for Campylobacter during 2014 and 15. And as you can see here, every year at an average of 280,000 cases related to Campylobacter are reported in the United Kingdom. And as you look at that, Campylobacter is one of the top priority organisms, and it has a high level drug resistance. And I'll be talking about just briefly about multidrug transporters. So multidrug transporters are classified into 5 major families, and you can see these are the single component system, and the one you see on the right side here, these are 3 component system. We call it as R&D transporter, and these are like a single component system. So just briefly, the role of this single component system, including the MAT transporter, which is the multidrug and toxin extrusion family transporters, they simply feed the antibiotics to the R&D transporter. What you see here, you see it takes the drug from the cytoplasm and then pump it to periplasm in gram-negative. And then from the periplasm, you can see these R&D transporter, they capture the drug and then they send it to external site. So having said that, in Campylobacter, people have identified CME ABC. There is a R&D transporter which involves in the extrusion of erythromycin, ciprofloxacin, norflux, as well as some dyes. Whereas there is no MAT transporter classified. Sorry, there is no transporter being studied extensively. So this is one of the R&D transporter we have characterized from a dangerous pathogen, Neisseria gonorrhea, and we have some understanding what how R&D transporters they work and how they transport substrates. So this is now the time to work on MAT transporters. So what are MAT transporters? So MAT transporters are secondary active transporters. They use either sodium or proton. Okay, so they are energy dependent transporters. So these energy dependent transporters are first characterized during 1970s, and then later in 1990s, people have identified the R&D transporters, which I mentioned, the triporate complex. So these MAT transporters, they transport cationic drugs and dyes and variety of molecules. And these transporters, they play a major role in cellular homeostasis. Okay, they transport all kind of toxic chemicals. These are classified into 3 major categories, subfamilies. One is called NOR-M subfamily, which is mainly present in most of the gram-negative organism, including Neisseria and Vibrio. And second category is DNF, that is DNA damage inducible family. And third one is eukaryotic MAT transporters. As I mentioned, eukaryotic MAT transporters are playing a vital role in our cellular homeostasis. They actually transport all kind of toxics from our kidney as well as liver cells. So what we know, these are like they have a general topology of 12 transmembrane helices. So the first half is similar to the second half. And what we don't know from the MAT transporters, because as I mentioned, in most of the pathogens, the MAT transporters are not well characterized. So what we don't know, how these transporters they work, what is the the mechanism of extrusion, and what are the substrate specificities. So we actually got a Campylobacter strain from one of the chicken farm in Scotland. Our collaborators, they isolated this strain, and then we looked at the MAT transporter by whole sequencing. So this is the MAT transporter. We call it a CC MAT. It's a MAT transporter from Campylobacter. And when we align with the other known transporter from other families, our MAT transporter is not actually having the well-conserved domains. As you look at these aspartic acid, and then again the aspartic acid, glutamic acid, or these acidic residues, or previously identified as a drug-binding pockets in the MAT transporters. These are actually not well conserved in our MAT. So what we did one of my collaborators, he actually produced a homology modeling, and thanks to him, and we found our MAT has a 12 transmembrane helices, as you look at here, and more likely the drug binding pocket should be in the N-terminal lobe, which I'm pointing here, which is transmembrane helix 4. Okay, what we did, we cloned the MAT transporter from C. coli, and then we performed the drug diffusion assay, which is disk diffusion assay here. Again, from the sequence I mentioned, one of the conserved domain, which is the tyrosine and then leucine, we mutated again this leucine. So what you see here, this is the control cell without MAT expression, and this is the mutant one. The one leucine 138 is mutated to serine, and that's the wild type. As you can see, the wild type conferred a very high level resistance to a number of antibiotics. As you look at, very importantly, it has shown resistance to erythromycin. As you look in here, it's like a 20 millimeter compared to the control one. So it's clearly, erythromycin is a main drug target, and I mean the drug of substrate. And by looking at our MIC data, as you can see here, it has 60-fold resistant to erythromycin and 30-fold to novobiocin, again 4 to 16-fold to different dyes and SDS molecules. So it's clearly erythromycin is one of the drugs recommended for egg-laying chickens. That's why probably we see high-level resistance. And we were looking at whether the CC MAT, is it a sodium or proton-dependent pump? So our data confirms our CC MAT is a proton-dependent pump. So what we did, this is a growth curve. What you see after 2 hours of growth in the presence of a drug novobiocin. And then what we did, we added CCP4. This is a protonophore molecule which interferes with the proton translocation. So after we add this molecule, you see the growth, the curve, it is flattened now. So it's clearly indicating that it is a proton-dependent transporter. Okay, again, what I'm showing here, these are some very interesting and intriguing data. And as I mentioned, we worked earlier in an R&D transporter called MTR-CDE. So from the understanding of the transporter earlier, we know how efficiently these R&D transporters, they transport drug across the molecule. Okay, so by looking at here, the drug molecule we used for this growth curve data is novobiocin, which is a cytoplasmic drug. And what you see here, you see by looking at the relative intensity of the growth in the presence of the drug, one can tell how efficiently these drug molecules are actually transported out of the cell. So you can see here. So we did same experiment, with C. coli MAT, and then we found this is equally important, equally efficient in transporting the drug molecules. So in that case, what it is, MAT is the major supplier of drug to R&D transporter. So they simply take the drug from cytoplasm onto periplasm that will be later captured by R&D transporter. So what I'm trying to say here is MAT transporters are equally important. At the same time, what happens is MAT transporter, they can only transport substrate to periplasm. And what happens if these molecules are not captured by R&D, what will happen? The cell can't cope with the excess accumulation of drugs in the periplasm. That means there must be some cooperative interaction or a synergistic kind of action between MAT and R&D transporter in order to efficiently transport these molecules out of the cell. Okay, so that's why we say MAT is, again, this MAT or the major supplier to R&D transporter. They supply all kind of drugs to R&D, and then R&D will be capturing and then send it out. So MAT is an important transporter, and it's not been characterized extensively in most of the gram-negative as well as the other pathogens. Just to summarize, as I mentioned, our C. coli MAT has, it's not having many of the conserved domains. It could be a subfamily of DIN-F cluster. And we noticed that it conferred high-level resistance to, novobiocin as well as, macrolide erythromycin. And we identified the key residue, which hasn't been characterized in any of the MAT earlier, which is the leucine and closer to the tyrosine, which could be a potential drug binding pocket in this group of MAT transporters. And from our data indicates these are like proton dependent transporter. And finally, I would like to conclude that as I mentioned, MAT or the major supplier of antibiotics to R&D. So the potentially future, we need to find a way to just to close the or how to break the chain, the supplier to the consumer. So again, finally, I'd like to thank my collaborators who actually, designed our the homology modeling for the new MAT, and then the other collaborators to help with us with the previous R&D transporter, and then our lab members, Tira, who cloned the MAT transporter, and then Romana, he performed all the drug efflux assays. Thank you for listening. I'm happy to take any question.
Brilliant. Thank you very much, Thamarai.